everyone, and welcome back to my channel. In today's breakout video, we are going to be taking a look at transcription and translation. Or, if we break that down even further, we're going to be looking at how DNA turns into protein. This is a topic that I do cover in some of my other videos, but I don't go into as much detail as I will be going into in this one. If you'd like my videos and the content, please make sure to subscribe and hit the little bell next to it to make sure that you never miss out on a new video. Let's take a look at the nucleus of a eukaryotic cell, as this is where the entire process begins. In the nucleus, we will find DNA. DNA is the genetic code of every living organism. Now, understand that DNA itself is very, very long and codes for many, many proteins, okay? But how do we get from the DNA to the actual protein? In order to go from DNA to protein, DNA, which is a double-stranded molecule, needs to be turned into RNA, which is a single-stranded molecule. This first process is known as transcription and occurs within the nucleus of the cell. Once this RNA is made, it is quickly processed into mRNA or messenger RNA. This messenger RNA then leaves the cell, and the next portion of the process actually occurs within the cytoplasm of the cell. Here in the cytoplasm, the messenger RNA will attach to a ribosome and be translated into protein. Both of these processes include several steps, and throughout the next few slides, we're going to be taking a more in-depth look as to how they occur. Let's first take a look at transcription, which is the process of going from DNA to RNA. Now again, remember that this process of transcription happens within the nucleus because that is where we are going to find the DNA. But now let's take a closer look at what exactly DNA and RNA are. Both DNA and RNA are nucleic acids. If you've watched my video on large biological molecules, you'll know that nucleic acids are polymers, which are made of monomers called nucleotides. Nucleotides themselves are made from a nitrogenous base, a sugar, and a phosphate group. Now, both DNA and RNA are nucleic acids that are composed of these nucleotides, but they do have a couple differences. One of them is that the sugar in DNA is deoxyribose, while the sugar in RNA is ribose. Another difference is in their nitrogenous bases. DNA has cytosine, guanine, adenine, and thymine as nitrogenous bases, while RNA has cytosine, guanine, adenine, and uracil. So you'll notice that all the nitrogenous bases are the same except for the fact that DNA has thymine while RNA has uracil. Let's get back to the process of transcription. With all that knowledge in hand, now we can understand that the process of transcription is going from DNA to RNA, or from nucleic acid to nucleic acid, with the difference that DNA has thymine and RNA has uracil. Now, DNA is a double helix, and in order for the process of transcription to begin, an enzyme known as RNA polymerase needs to bind to the DNA. It does so at a specific region known as the promoter region. Now, there are various promoter regions throughout the entire DNA that are at the beginning of many different genes. Once the RNA polymerase binds to this promoter region, it's going to open up the two strands of DNA, and it's going to allow the RNA nucleotides that are complementary to the DNA template strand to start to bind. This is going to allow the RNA to be made. I'm going to draw out the two strands of DNA straight so that you can better see how this process occurs, but understand that DNA is helical. So one strand of DNA runs 5' to 3', while the other strand of DNA runs from 3' to 5'. This top strand is known as the non-template strand, while the bottom strand is going to be the template strand that is used for RNA synthesis. Once the RNA polymerase has bound to the DNA at the promoter region, this is known as initiation, then the RNA nucleotides that are complementary to the DNA template strand can start to be added. This RNA strand is going to be made using the template strand of DNA, which is running from the 3' to 5' direction. In this case, the RNA is going to be synthesized in the 5' to 3' direction, which is the direction which we read these nucleotides. The RNA polymerase will continue to move downstream, elongating the RNA transcript. Eventually, the RNA transcript will be released, and this is when termination occurs. The polymerase will then detach from the DNA itself, and we will now have our RNA strand. For those of you who may be interested in how the nucleotides for the RNA pair up to the DNA, we're going to take a closer look at that. So in this slide, we're going to take a look at the same process that I just spoke about, except this time we're going to be looking at the nucleotides, specifically the nitrogenous bases that match up. So the first thing again is we want the RNA polymerase to bind to the promoter region in order to open up the DNA so this process can occur. If we look closely at the nucleotides within the DNA, we'll notice that they're arranged in such a way that A, or adenine, matches up with T, or thymine, and G, guanine, matches up with C, cytosine. So in this arrangement, we have the nucleotides for the non-template strand, and then the complementary nucleotides that are on the template strand. Now the 3' to 5' strand is going to be our template strand from which the mRNA is going to be made. As the RNA polymerase moves down the DNA strand, it is going to attach the correct nucleotides to make the new RNA molecule. So let's take a look at what this looks like. Remember that our RNA is going to be made from the template strand. So let's look at this example and figure out what's going to happen. So in this case right here, where we have a T, first in the RNA would then be an A. Next, if we look at the next nucleotide, it's an A for the DNA, which means for the RNA, it's going to be a U, or uracil. Remember, in RNA, there is no T or thymine, so instead, it's going to be a U. Next on the DNA, we see a C, so for the RNA is going to be a G. Next, we see a T, so again, we're going to have another A. Then we have an A, so it would be a U for the RNA. Next, we have two Gs, and so both of those would be Cs on the RNA. The two Ts would then be two As. And then finally, the last three, A, G, G, would be U, C, C. This would continue on until the entire RNA was encoded for. Now I just did a little piece to show you an example of how this works with the different nucleotides. Once this RNA is made from the 3' to 5', it's actually being made in the 5' to 3' direction. Now all nucleic acids are read from 5' to 3', so this is the correct orientation, and it's going to be made in the opposite direction from its template. So the template, again, you see is in the 3' to 5' direction. As the RNA polymerase puts those nucleotide bases on there, it is actually making the RNA in the 5' to 3' direction. After this process, we now have our RNA. We're still in the nucleus of the cell, but we have to go from RNA to mRNA. So what's the difference and how does this get processed? I do talk about this in my video, What is mRNA? But briefly, when RNA is first made, it is referred to as a pre-mRNA because it contains both introns and exons. The introns will be removed by RNA splicing as they will not be part of the final mRNA product, while the exons will go back together in order to create this mature mRNA. Once this mature mRNA molecule is made, it is ready to leave the nucleus through the nuclear pores and enter into the cytoplasm. Once the mRNA is in the cytoplasm of the cell, it can bind to a ribosome where translation will take place. The process of translation includes 3 types of RNA. First, we have the mRNA that we've been talking about so far. We also have the ribosome, which is rRNA. And then finally, we're going to introduce the tRNA. The ribosome itself has 3 binding sites: an amino acid site, a polypeptide site, and an exit site. The AUG is the start codon of the mRNA, so the first part of the mRNA is actually not even translated. What happens first is that the tRNA molecule will come in and bind to AUG. The mRNA is actually read in triplets or codons, and the tRNA triplet code is referred to as the anticodon. The tRNA anticodon will match up to the mRNA codon and deposit the appropriate amino acid. In this case, A from the mRNA will match with U, the U will match with the A, and then the G will match with the C. The first amino acid will be dropped off. The first amino acid is always going to be methionine. This process is going to continue on where another tRNA anticodon is going to come in and match with the next codon on the mRNA. Each tRNA anticodon always carries the same amino acid. The amino acid that is carried is determined by the anticodon on the tRNA. In this way, the same protein can always be made by the same mRNA code. This is a very specific process. As the tRNAs continue to come into the ribosome, it will continue to build up the polypeptide chain by the addition of amino acids. The tRNA will enter into the amino acid site, then it will move to the polypeptide site where the amino acids will be joined together, and then it's going to exit through the exit site. So you can see it's coming in through the amino acid site. The chain is going to be made in the polypeptide site, and then the tRNA that has now lost its amino acid is going to exit through the exit site. So the chain is being built up in this polypeptide site. Again, as I had mentioned before, this is going in the 5' to 3' direction on the mRNA. So the ribosome is going to move through in the 5' to 3' direction as the chain is growing. Eventually, the tRNA will reach a stop codon. Once this stop codon is reached, the polypeptide chain or protein will be released. There are 3 different stop codons or termination codons that can be found on the mRNA. These include UAA, UAG, or UGA. These are codons that the tRNAs do not recognize, and because of this, allows for the release of the protein. Once the protein is released, it can be folded correctly, have any post-translational modifications done to it that may be needed for it to work properly, and then potentially escorted to do its job. This varies depending on the proteins made. In the next slide, we're going to take a closer look at this process using the example that I started in the previous slide. Let's quickly revisit the example that we've been looking at. So here we have the DNA. Here's the template strand down here. And then here is the RNA that had been made from that template strand. So this was transcription that occurred within the nucleus. And now we're going to take a look at translation that occurs in the cytoplasm, specifically on a ribosome. So in the previous slide, I described how translation happens. In this slide, we're going to take a look at how specifically the tRNA anticodon binds with the codon of the mRNA. So first, let's take a look at what the tRNA anticodons would be. Say you have to do this for a class, you're trying to figure this out, and then I'm going to show you how to figure out the amino acids. So it's read in triplets, so we're going to read them by threes, and then we're going to figure out what the tRNA anticodons would be. So the first one is AUG. We just did that in the other slide. So this would be UAC for the tRNA anticodon. The next triplet is AUC on the mRNA. So this would be UAG on the tRNA anticodon. The next triplet is CAA, so the tRNA anticodon would be GUU. And then finally, the last one in my short example here is UCC on the mRNA, and for the tRNA anticodon, that would be AGG. So this gives you an idea of how to figure out what the anticodon would be that would attach to that mRNA. Now I started my example at the start, and I just went on for a few so that you can kind of get that idea. Now, how do we figure out what the amino acid would be for each one? And that's what we're going to do now. In order to figure out which amino acids are going to be put in for the different codons of the mRNA, you're going to need a chart like this. So in your book, it may be in this wheel form, or it may also be in table form. You can easily look at these up on the web as well. The confusing part about this is that these charts are referring to the codons on the mRNA. And so sometimes you may be asked to look at or find the tRNA anticodons, which we just did. But in order to find the amino acid using these charts, we're actually going to go back and look at the mRNA itself. So let's take a look back at the mRNA, and our first codon is AUG. If we use this chart to find out the amino acid, we start from here. This is the 5' to 3' area. So we're looking for AUG. So here's our A, here's our U, and here's our G. So AUG brings us to methionine, which, as I mentioned before, is always the first amino acid. So I'm going to come back here and just put the abbreviation for methionine here. That's our first amino acid from that AUG. Now let's look at our next set. Remember, again, we're using the mRNA code to do this. We have AUC. So now we're going to take a look for the AUC. So again, we're going to start here in the middle with our A. Here's our U, and then here's our C. So AUC codes for isoleucine, or ILE for abbreviation. Our next codon is CAA. This time we're looking for CAA. We'll start in the middle again. Here's C, here's our A, and here's our other A. So CAA is for the amino acid glutamine, or abbreviated GLN. And let's go ahead and find our final one, which is UCC. Again, our final one is UCC, so we're going to start in the middle. Here's the U, here's our C, and here's our other C. So UCC is for the amino acid serine, or it's abbreviated SER. This concludes our example. It's a small example, but hopefully it gives you the idea of how we go from DNA to mRNA, and then how we find the tRNA anticodons, and then finally how those amino acids are found using that mRNA codon chart. Thank you so much for watching my video. If you like this video and want to see more, make sure that you subscribe, Click that little bell next to it to turn on notifications so you never miss out on a new video. Thank you.